In this video tutorial, I am going to discuss about alpha oxidation of fatty acids. Remember that beta oxidation is the most predominant, or you can say default pathway for fatty acid oxidation. And this beta oxidation requires clean, unmodified beta carbon. But in some fatty acids, beta carbon is modified, mostly containing methyl group, which is not suitable for beta oxidation. So, in such cases, fatty acid requires alpha oxidation. Let's understand this with the classic example of phytanic acid. And this is the structure of phytanic acid. So as we can see, phytanic acid has 16 carbons in its backbone and 4 methyl groups attached as a side branch. Because of these side branches, it is classified as a branch chain fatty acid. Further, this is the terminal carbon. So this is alpha and this is beta carbon. This beta carbon contains methyl group. So as I stated earlier, as beta carbon is modified with this methyl group, this cannot undergo beta oxidation. So this phytanic acid must utilize alternative oxidation pathway, and that is alpha oxidation. And one important point, where does this alpha oxidation happen? It occurs in the endoplasmic reticulum and peroxisomes. This is in contrast to beta oxidation, which occurs in mitochondria. Now let's see how alpha oxidation occurs step by step. So let's focus on this alpha carbon and terminal carbon. Now just like beta oxidation, here also activation of fatty acid is required. So accordingly, this phytanic acid also undergoes activation. In this reaction, coenzyme A is added into the fatty acid. So this terminal COO is converted to C double bond O bound with SCOA. Remaining fatty acids, including this alpha carbon, remains unchanged. So this alpha CH2 and this remaining fatty acid. The name of this activated phytanic acid is phytanoyl coenzyme A. This reaction requires energy. So ATP gets converted to AMP and pyrophosphate. So essentially, two high energy phosphate bonds are utilized in this activation reaction. Now, some textbooks wrongly mention that alpha oxidation does not require activation of fatty acid, but as we had just seen, activation is actually required here also. So anyways, once the fatty acid is activated to phytanoyl CoA, it undergoes alpha oxidation, which is a multi-step process. And here, phytanoyl CoA hydroxylase is one of the important enzymes. I will not go into the minute details of these steps, but the ultimate effect of this alpha oxidation is that this alpha carbon gets oxidized. So this bond gets broken and it releases this carbon as a carbon dioxide. And remember, this carbon was originally this carbon and this bond was originally this bond. So in this original structure of phytanic acid, we can say that by alpha oxidation, this bond gets broken and this terminal carbon gets released as a carbon dioxide. After removal of this carbon becomes terminal carbon. So this becomes alpha and this becomes beta carbon. As this beta carbon is free of methyl group, it can now undergo beta oxidation. So this bond gets broken. So this 3 carbon fragment gets released as a propionyl coenzyme A. Once this gets removed, this carbon is new terminal end. So this is new alpha carbon and this is new beta carbon. Again, this new beta carbon is free from methyl group. So it goes into beta oxidation. So this bond gets broken and these 2 carbon fragments get released as a acetyl coenzyme A. So now this becomes new terminal carbon, this becomes alpha and this becomes beta carbon. Again, it is free from methyl group. So one more beta oxidation takes place and this bond gets broken, releasing this 3 carbon compound. So it is released as a propionyl coenzyme A. So now this is new terminal carbon. So this becomes alpha and this becomes beta carbon. Again, this is free from methyl group. So beta oxidation occurs, leading to breaking of this bond and release of this 2 carbon fragment. So it is released as acetyl CoA. After removal, this becomes new terminal carbon. So this becomes alpha and this becomes beta carbon. Here also beta carbon is free from methyl group, so beta oxidation occurs and this bond gets broken, leading to release of this 3 carbon fragment. So it gets released as a propionyl CoA. Now this carbon becomes new terminal carbon, so this becomes alpha and this becomes beta carbon. Here also beta carbon is free from methyl group, so beta oxidation can occur, leading to breaking of this bond. So this 2 carbon fragment and this 4 carbon fragments get released. This 2 carbon fragment is released as acetyl CoA and this 4 carbon fragment gets released as a methyl propionyl CoA. So as you can see, in case of phytanic acid, there is initially only one alpha oxidation cycle followed by subsequent 6 beta oxidation cycles. And if we tally the products of complete oxidation of phytanic acid, we find 1 molecule of carbon dioxide, 3 molecules of propionyl coenzyme A, 3 molecules of acetyl coenzyme A, and 1 molecule of methyl propionyl coenzyme A. Now let's bring this back to the clinical world. There is a rare genetic disorder called Refsum Disease. In this condition, the enzyme phytanoyl CoA hydroxylase is defective. Remember, that was the key enzyme in alpha oxidation. Because this enzyme doesn't work, phytanic acid cannot be processed. So it accumulates, and it accumulates especially in tissues that have the highest lipid content and metabolic activity. That means the retina, the myelin sheath of nerves, the skin, and the heart. Let's go through each one. In the retinal cells, phytanic acid gets incorporated into cell membranes. This alters the membrane fluidity and stability. Over time, the retinal cells degenerate, leading to a condition called a retinitis pigmentosa, where the patient experiences night blindness and progressive tunnel vision. Now, this myelin sheath is the protective and insulating lipid layer surrounding the nerve fibers. Abnormal accumulation of phytanic acid in this leads to destabilization of myelin structure, leading to a process called demyelination. This slows down or blocks the transmission of electrical signals along the nerves, causing the peripheral neuropathy. It has a symptoms like weakness, numbness, and loss of reflexes, which is called as a areflexia. Now, in the skin, the outermost layer of the skin, the stratum corneum, acts as a barrier. Phytanic acid accumulation leads to disruption of this barrier, so it cannot retain moisture properly, leading to excessive dryness and the characteristic scaling known as ichthyosis. And in the heart, cardiac cells generate the electrical signals that drive a normal heartbeat. But accumulation of phytanic acid in cardiac cell membrane leads to loss of this function. So heartbeat can become irregular, and this condition is called as arrhythmia. This is the most immediately life-threatening complication. So to summarize, alpha oxidation is a specialized oxidation pathway for branch-chain fatty acids like phytanic acid, where the standard beta oxidation is blocked by methyl group at the beta carbon. Alpha oxidation removes that blockage by eliminating the terminal carbon as a carbon dioxide, after which beta oxidation can proceed normally. When the key enzyme phytanoyl CoA hydroxylase fails, phytanic acid accumulates in the body and causes Refsum disease, affecting the retina of eye, nose, skin, and heart. I hope the concepts are clear. If there is any doubt or confusion, please drop it in the comment section below. Thank you.